Today we are exploring the essential high-yield lab techniques used in cell biology. Let's unlock the secrets of how scientists study life at the microscopic level. First up is microscopy. It is critical to distinguish between magnification and resolution. Magnification simply increases the apparent size of an object, while resolution determines the clarity and amount of structural detail you can actually see. Since cells are mostly water, they are usually transparent. Staining utilizes specific chemical dyes that bind to specific organelles, creating visual contrast so we can easily spot structures like the nucleus or cell wall. When scientists want to isolate specific parts of a cell, they use cell fractionation through centrifugation. Cells are blended open and spun at incredibly high speeds. The heaviest parts, like nuclei, settle first, followed by smaller organelles as speed increases. Tissue culture involves keeping cells alive and multiplying them inside a lab, completely separate from the original organism. This sterile environment is incredibly important for investigating cancer therapies, genetic engineering, and producing vaccines. Next is chromatography. This technique separates chemical mixtures based on how fast they move across a stationary medium. It is widely used in botany to isolate and identify distinct photosynthetic pigments like chlorophyll A and B. Electrophoresis applies an electric current to push charged biological molecules through a special gel matrix. Because smaller molecules navigate through the gel matrix much faster than larger ones, it cleanly separates chunks of DNA, RNA, or proteins by their size. Spectrophotometry is used to determine exactly how much of a substance is present. By passing light through a solution and calculating how much light is absorbed, scientists can compute precise chemical concentrations instantly. Finally, we have microdissection. This ultra-precise technique uses specialized microscopic instruments, or even microscopic lasers, to physically cut out and isolate a single cell from a tissue sample for deep genetic analysis. And that is your Prep N5 masterclass on cell biology techniques. If this breakdown helped clear up your lab chapters, please click that subscribe button, hit the notification bell, and give this video a thumbs up. Thanks for watching, and keep studying smart.